Gene expression analysis is a powerful tool for measuring biological processes that underlie disease progression and therapeutic effectiveness. However, analyzing RNA using traditional technologies requires complex steps to convert RNA to cDNA, which is then amplified and quantitated as a proxy for the original RNA. This process introduces variability which may lead to biased data and an inability to replicate results. Why introduce and count cDNA errors when you can simply count the native RNA? Direct detection using nCounter technology provides highly robust data across clinically relevant samples, while reducing hands-on time and simplifying analysis. nCounter technology gets you to reproducible results faster. Using molecular barcode technology, RNA is directly tagged with a capture probe and a reporter probe that are specific to the target of interest, creating a unique target probe complex. After hybridization, excess probes are removed, leaving only purified target probe complexes. These complexes are immobilized and aligned on the imaging surface. The sample is then scanned by an automated fluorescence microscope, where labeled barcodes are directly counted and the data analyzed through an intuitive analysis software. Molecular barcoding technology can be used with RNA, microRNA, protein, and DNA analytes. Direct detection with nCounter technology provides several advantages: highly reproducible data, robust performance on FFPE, single-tube multiplexing, simple protocol with minimal hands-on time, flexible sample input, and simple data analysis. Faster than NGS, simpler than qPCR.
The NEB-Next Ultra 2 Directional RNA Library Prep Kit is the next generation of strand-specific library construction kit, compatible with Illumina sequencing. It enables production of high-quality libraries from low nanogram to microgram input amounts of total RNA using a streamlined workflow. Before library construction, ribosomal RNA must be removed by either of two methods compatible with the kit: poly-A mRNA enrichment or ribosomal RNA depletion. After removal of ribosomal RNA, the RNA of interest is fragmented by incubation at high temperature in the presence of divalent cations, followed by hybridization to the random hexamer primer for reverse transcription. Next, first-strand cDNA synthesis occurs. A reverse transcriptase extends from the hybridized primer along the length of the RNA molecule. Actinomycin D is added to prevent second-strand synthesis in this step. Second-strand synthesis then takes place. Strand-specific library construction is different from standard library construction in this step, as uracils are incorporated into the second strand, and this gives the protocol its name, the DUTP method. After a cleanup step, the double-stranded DNA molecules are end-repaired to create blunt phosphorylated ends, and a single adenine overhang is added to the 3' end. This enables efficient ligation to adapters with thymine overhangs. These adapters can be the hairpin loop-shaped NEB-Next adapter or other Illumina sequencing-compatible adapters. The uracil-containing strand is then selectively removed with a user enzyme, which is a mix of UDG and Endo-8. If applicable, user also opens the hairpin loop of the NEB-Next adapter. What remains is a single strand in the final library, providing the library's strand specificity. Next, either a cleanup for smaller fragments or a size selection for larger fragments is performed. The final step is PCR amplification of the library using a Q5 high-fidelity DNA polymerase master mix with the recommended number of cycles based on input amount. This step selects for molecules with an adapter at each end, increases library yield, incorporates barcodes to enable multiplexing, and incorporates P5 and P7 sequences required downstream in the sequencing process. After a cleanup, the library is ready to move to cluster generation and sequencing.
Through our platform, we integrate clinical, molecular, and outcomes data, and make that data accessible and useful, providing tools and insights to better understand a patient's disease. We have built a highly robotic next-generation sequencing lab, offering a variety of integrated DNA and RNA sequencing capabilities, including whole exome and large panel DNA tumor normal match for solid tumors and hematologic malignancies, along with our cell-free DNA assay. In our CAP-accredited and CLIA-certified lab, we're able to provide high-quality sequencing services at industry-low pricing to ensure every patient who should be sequenced has access.